Okay, well, I want to thank you, Shane, for coming back and giving us a talk today on your work on palette fusion and disruption. Really appreciate you coming back. I know this was a popular talk in the past, and I'm looking forward to hearing it again. And hope that we'll catch up on your other work at Vanderbilt at some point as well. So I'll turn things over to you and appreciate you being willing to talk.
All right. Thank you, James. Yeah, I'm a professor of physics and biological sciences at Vanderbilt University. I'm going to tell you about some work we did modeling secondary palate fusion and how it gets disrupted by chemicals like dioxin, for example, during development. And this is work I did in conjunction with Tom Knudsen, who's on the call here, during a sabbatical year I spent at the EPA in Research Triangle Park. So their grant support was critical for this, and so was the Oak Ridge Institute for Science and Education. I want to make sure I mention them. All right, so what were we trying to do, and what did we get out of this model? So there's a paper published on it in Chemical Research and Toxicology. I invite you to go look at that. That'll give you all the details or any background you want to know about this. But what we were trying to do is this: to take CompuCell3D and handle both sort of the cellular mechanics, how the cells interact with one another for multiple different cell types. And we'll get into all the different kind of cell types that we have there. And think about how the palate shelves grow towards one another, meet, and then fuse with one another. And we're going to have this large set up here in the top left, this set of bioregulatory networks that's going to control how the cells behave in the model. All right, so it's a cell-based model of palate fusion. It incorporates all of this biochemical regulation. We can do some in silico genetic knockouts, and those match up to what you actually see in experiments. We'll talk about all those in detail. And then we can mimic what happens when you add a molecule like dioxin in that interferes with specific pathways in this regulatory network, and think about how that affects palate fusion and disrupts palate fusion under some circumstances. Okay. So tissue fusion is something that Tom and I were thinking a lot about during that year. We decided on the palate as a sort of canonical example of this, but let me explain what happens with the palate and what happens in all these other things. So if you look up here at the top right, this panel labeled B, this is from a mouse at embryonic day 13 and a half, and the two shelves of the palate are growing down beside the tongue. T there, and the shelves are labeled P. They flip up above the tongue and then grow towards one another. You can see that at embryonic day 14. And by embryonic day 16, they've met, and there's a little bit of a thin line between them that really is an epithelial tissue that's caught between the two. Right? And the key to tissue fusion is that you'll have two groups of tissue that have some mesenchyme underneath, epithelial layers on the outside, and you're going to fuse them together. And to do so, one of the things you have to do is get rid of that epithelium that's separating the two, right? And have a single epithelium around the joined tissue. So we're looking at this in detail in palate. Similar things happen over here on the far left in cardiac development, all the septa of the neonatal heart. In neural tube fusion, they're in the middle, optic cup, diaphragm, urethra. Right, there's lots and lots of examples of this that happen during development. This idea that two tissues growing towards one another have to fuse into one tissue. And we know a lot about the mechanisms here, and cell adhesion plays a role, apoptosis plays a role, epithelial mesenchymal transitions, migration signaling, and we're going to try to incorporate all of that. That's one of the powers of CC3D, is it allows you to put all of those sort of things into a model. All right, so in detail, here's some close-ups of what the cells actually look like during palate fusion. So here's those palate shelves, PS, growing towards one another and meeting at the midline. And you can see that they just touch, and they're in the middle. You can see the darker brown area is the epithelium. The bluer stained region is the mesenchyme underneath. And that medial epithelial seam, that's what this MES is, has to dissolve over time so that you get a single confluent tissue. You can see this seam in the cross sections we're looking at, or if you look at a top-down view of the mouse palate, the upper palate in C, and then eventually in D, you can see as the palates fuse, the palate shelves fuse, you end up with just these little blue dots, these little pearls left in between that eventually go away, right? Those are epithelial cells that are sort of trapped in between the two shelves as they're trying to fuse, but they will disappear over the next day or so. All right, so that's what we want to try to model and understand and gain some insight for. Just another view of it. I think we've touched on this enough that we can go into it. So outline. I'm going to sketch the computational model, what biophysics we try to include, what biochemical regulation we try to include, show you some simulations of wild-type fusion and in silico knockouts, simulate a toxicants effect, in particular dioxin. Then we'll look at the model code a little bit, and there's some exercises built into the end of this that you can work on. So here's a close-up of the output of our model, and so I can introduce the players to you. So we have over here on the left, you can see the two palatal shelves that are going to grow towards one another, and they're going to meet in the middle here. And so this is after just about 1,200 Monte Carlo steps of them growing in the model. The nasal side is up top, the oral side is below, and medial is what we'll call in the middle as they grow towards one another. On the right is a label of all the different cell types that we have in the model. So there are mesenchymal cells in this light cyan. The mesenchymal ECM is modeled in this sort of brownish yellow. And so it's modeled as a cell in CompuCell, even though it isn't one, but we take advantage of that framework for putting it in. There's an explicit basement membrane. There's basal epithelial cells there in a blue, and then what's called a periderm, which is a very squamous epithelium on the outside of the palatal shelves that's in that pink and that purple. And what you'll notice is that periderm in particular, the reason we have pink and purple there are those cells have apical and basal compartments, and that'll be important for polarizing the epithelium and making sure it stays polarized during the model and during the simulation. Right, and as you've been learning from all this stuff on CompuCell3D, right, each cell occupies multiple points on a hexagonal lattice, and you can kind of see that when we take a close enough look, and you can see the pixelation of the cells that are in there. All right, so those are the players. The periderm, basal epithelium, basement membrane are our epithelium, and that's what's going to have to go away and get out of the way for the mesenchymes to fuse when these palatal shelves meet one another. I have a model. No, not a movie yet. Okay. So the terms we use for each of the cells in there, CompuCell3D is a POTS model, Metropolis Monte Carlo algorithm for running through that. We have terms for the target area of each cell, a target perimeter of each cell, and a target length for some cells. And that combination lets us specify sort of the shape of cells, of cell types. And then obviously we have the contact energy between all the cells and between different cell types. This is our matrix of contact energies that we have. And what I will say is, as you get used to coding these models, the exact values here are not critically important, and I don't have experimental evidence for exact values for any of these, right? But I can give you an idea of what the ordering of them should be, what the hierarchy of these numbers should be, and I can give you some examples of that. So for example, if we look at this little 2x2 matrix here in the middle that says how the mesenchyme interacts with the matrix, the mesenchymal cells interact with the matrix in the mesenchyme. Notice that the highest energy you see among those four is for a mesenchymal cell contacting another mesenchymal cell. So high energies are unfavorable, so the cells aren't going to clump together. And instead, the lowest energy is for a mesenchymal cell with the matrix, and so cells are going to surround themselves with matrix. And matrix is sort of neutral, interacting with itself. And so that's going to give us cells that are dispersed within this extracellular matrix for the mesenchyme. If we look instead at what's going on with the epithelium, right? In particular, look at how, right, each of the things interact with the medium, the black stuff that's out here, right? It's the surrounding medium. If you look across those energies, the lowest is the apical part of the periderm cell. So that's what's going to be in contact with the medium. And then those apical periderm cells contact one another pretty well, and then contact the basal part of the basal part of the paraderm. The 30 with the parentheses 2 just means that the contact energy of paraderm A with paraderm B is 30 if they're parts of different cells, but 2 if they're part of the same cell. So the same cell sticks together really well then, because it has a low energy favorable contact for those two. And you can go through and look at the ordering of this, and it's going to sequentially say, all right, paraderm cells like to be in contact with basal epithelium. Basal epithelium likes to be in contact with basement membrane, and then the basement membrane is in contact with the matrix underneath, right? So it's the hierarchy of energies that you can really think about specifying the behavior of cells in the model. And then we wanted to put in the biochemical regulatory networks for this. So this is the whole picture of it, but there's a couple of modules in here that really become important for it. And I will highlight some of those modules. So one of them is this loop right here between sonic hedgehog, SHH, and fibroblast growth factor 10, FGF10. And what's on the arrows is what mediates the interaction. The labels on the arrows aren't really in the model, right? The model is just the interaction between these two morphogens or signaling molecules. And sonic hedgehog secreted from the medial epithelium induces the mesenchymal cells to secrete FGF10, and FGF10 secreted by mesenchymal cells induces the epithelial cells to secrete sonic hedgehog, right? So we have this feed-forward feedback loop. And so if one of these starts to go up, it drives the other one up. And so you link the growth of the mesenchyme to the growth of the epithelium by having this signaling back and forth between them. And then in contrast, well, I should say we got a little bit extra here. BMP2 also plays a role in the mesenchyme, is linked to this in proliferation and matrix secretion by these mesenchymal cells. And FGF10 also induces proliferation of cells out here in the epithelium. So not only does FGF10 increase sonic hedgehog production by the epithelium, but it increases proliferation of the epithelial cells itself. And then the other part of this, it's modular, is this interaction between epidermal growth factor, EGF, and transforming growth factor beta, TGF beta 3. And notice that this is a mutual inhibition in this case. And what that's going to give us is a situation where one or the other goes high, right? It's a switch. Either EGF goes high and drives TGF beta low, or vice versa. And then those differentially control apoptosis, EMT, motility, and proliferation. And you'll notice that what happens is if TGF beta goes high, the cells in the seam, the epithelium, apoptose, undergo transitions to mesenchymal, and move a lot. But if EGF goes high, all of those things are suppressed. And so this switch is going to control whether the seam dissolves between the two palates as they approach one another. And I think there's other things that are in the model, but those are the really key parts of it. The other ones are really sort of to match some of the gradients we see oral to nasal within the palate shelves, but that's small details. And I will say, if anyone has questions, feel free to speak up or to put them in the chat as we go through this. I'm happy to stop and answer them as we go across it. All right. So here's a movie of the simulation, and the shelves are going to grow towards one another, and then you're going to see the epithelial cells that get caught in the seam initially, then migrate, apoptose, transition to mesenchymal cell types. All of those things take place and clear out this mesenchymal seam. So here's the movie playing. I hope that the frame rate doesn't get messed up too bad by Zoom. I'll let this loop back and we'll show it again. You'll notice some of those pearls of epithelial cells that get stuck there and then to change over time. And the little red cells that are left are some of the epithelial cells that we marked that became mesenchymal cells. And we just turned them red so we could see them, see how often that happened. And then there's still frames over there on the left for that. Okay. So that's what we intend to happen. That's what should happen in the wild type palate shelves. They meet and fuse with one another. And we can look at that movie again, but now also highlight all these fields we have for the signaling molecules. Remember that the top two up here, SHH and FGF10, this is the feedback loop that coordinates growth of the mesenchyme and the epithelium. And then down here, the bottom row, TGF beta and EGF, this is the mutual inhibitory switch that controls whether the epithelial cells apoptose, migrate, get out of the way. So you can see how these fields change with time over the course of the simulation. And notice in particular, if I scroll back through this, when the shelves first meet, we're at a stage where TGF beta is high. So it's keeping, it is now starting to transition so that these cells are going to apoptose and migrate out of the way. And you can see this forward feedback loop between sonic hedgehog secreted by the epithelial cells and FGF10 secreted by the mesenchymal cells. All right. So if we look in detail and compare this to what we see in experiments, this is marking sonic hedgehog from an experiment and comparing it to what we have in the model. So it's in the epithelium and in particular on the oral side of the epithelium as we have it here. Rotate this a little bit so we can match it up. A couple of different stains that were used to look at it, but they're in about the same pattern as we have in the model. FGF10, here's the experimental panel for it and the model panel for it. BMP4 is a little harder to see, but it's the darker blue stain that's sort of throughout the mesenchyme there, and that's where we have it in the model. FGF7 is more on the nasal side of the mesenchyme, where it is in the model. Noggin, which is a modulator of the TGF beta signals, is mainly on the oral side of the palate shelves. And there's TGF beta that's coming up from the epithelium there in the middle. So the wild type model was set up so that it reproduced the patterns of these signaling molecules appropriately. And it did that by specifying which cells secrete them and how that secretion is regulated by other secreted molecules and by the environment of each cell, right? Is it exposed to other epithelial cells? Is it buried in the mesenchyme? These sorts of things control which ones secrete which chemical at which time, right? It's just a list of rules that are in the model. Yeah, and this was just highlighting how those two work together. And you can see that both of them stay high for a long time as the shelves are growing. And as the shelves then meet, then they start to decrease because we're not having that much growth anymore. And if we look at the TGF beta, you can see it going away as the seam disappears there. So one of the important checks we wanted to do on this is, okay, we've got the model, the fields look approximately right, the cell behaviors look approximately right. What happens if we start knocking out particular pathways that are present in the model? Does it reproduce what we expect to see from experiments that have been done? And so if you knock out sonic hedgehog in the model, the shelves don't grow. That's what happens when you do the experiment in mice as well. You knock out BMP2, the shelves don't grow very well, although there's some misregulation of the epithelial layer and it overgrows a little bit. FGF10 and BMP4 there in the middle have some effect on growth of the mesenchyme, but it's enough that the shelves can still meet, and eliminating FGF7 or noggin really doesn't have any effect. These are sort of making small changes in what's happening nasal to oral in the palate shelves. So they're not having a huge effect on it. And this is what you see when you do the particular knockouts in mice as well. 3 more in silico knockouts we can look at that have become more interesting for thinking about what dioxin does down the road here. If we eliminate TGF beta signaling, then the shelves meet and the epithelium keeps on proliferating. And we get this gross buildup of epithelium between the shelves, and they're not really allowed to fuse. And what will happen is then later in development, as the shelves continue to grow, because they were never able to fuse, this epithelium will pull back apart. Right, this sort of block that's in the middle there. EGF mutant doesn't have much of an effect, and surprisingly, it doesn't have much of an effect in a mouse model when you knock it down. And MMP9, matrix metalloproteinase 9, does leave a little bit of the seam in between, and that's what you see when it's knocked down, right? That it tends to increase the duration of the seam between the palatal shelves. So then linking the model to toxicology, right? I was working with folks at EPA. We're interested in how environmental chemicals impact this. We looked in particular at dioxin as a class of chemicals that activates the aryl hydrocarbon receptor. And what dioxin does in terms of thinking about the adverse outcome pathway for this is you can see this arrow down here. It goes to the aryl hydrocarbon receptor, and that modulates TGF-beta signaling. And because it modulates TGF-beta signaling, it affects apoptosis, proliferation, and differentiation in those epithelial cells, changes how the medial edge degenerates, whether those epithelial cells get out of the way and allow fusion, alters fusion, and eventually leads to cleft palate, right? So that's the pathway that we think this is working within. And Nisha Sipes at EPA helped us put this together. Very nice job from Nisha there. And then we wanted to see if we could model this in the model that we had that we were building. Now, one of the keys is, all right, we're going to put a chemical disruptor into here. What parameters, what knobs in the model should this affect, right? Where should it go? And so looking and digging into the literature and that adverse outcome pathway that I just showed you there, aryl hydrocarbon receptor affects this EGF-TGF-beta switch. And we implemented it here as the aryl hydrocarbon receptor leading to increased production of EGF. And because EGF gets increased production, it throws this switch towards EGF that blocks apoptosis, epithelial mesenchymal transitions, and motility, and prevents the seam then from getting out of the way, right? From dissolving and letting the palate shelves fuse. That's the idea. So we implemented this in the model. And a couple of things of note when we think about this sort of mutual inhibitory switch that we're going to put in biochemically, depending on the parameters that we put in there for it, and we don't know what exactly the parameters of it should be, we can come up with two classes, right? One is this one that's in panel B that I'll call a low hysteresis switch. So you notice that as we change the production of EGF receptor, at low EGF receptor, TGF-beta is the one that wins the switch competition. And at about 1.2-fold EGFR, it switches pretty quickly to EGF being the one that wins the competition. And there's not a lot of hysteresis in here, right? It doesn't extend the TGF-beta line for too far before it switches over. But in C, depending on the parameters, you could also have a high hysteresis switch where you start as before with high TGF-beta, and at the switchover point where EGF is thermodynamically more stable or is the winner, you can still have pretty metastable TGF-beta 3 being high, and that extends for a long, long ways out here in terms of EGF receptor fold change. And so this is a high hysteresis switch. It means if TGF-beta 3 was already high, it can stay high for a long time before it eventually switches over to EGF being high. And if EGF had been high, it can stay high for a long time before it would switch over to TGF, right? It's resistant to making the switch, so it will go past the threshold at which it normally would happen. Okay. So we'll look at both what both of those would do to the model under different circumstances. So here's the low hysteresis switch when we have constant exposure to something like dioxin, constant exposure that's leading to aryl hydrocarbon receptor activation, and the low hysteresis switch, so a pretty clean switch between high TGF-beta or high EGF. And if we let this run, you'll see that there's a little bit of delay in the disintegration of the seam, but it eventually goes away, and these fuse okay. Right, we have some of these pearls left in between that are residual for a bit there. Now, if we switch instead to the high hysteresis switch, right? So now this TGF beta was already high because of the production, but we're going to try to switch it to EGF high because we have this exposure. And it's just more resistant to that, right? It's harder to get rid of those cells in the seam because there's a lot of hysteresis in the switch. Even though EGF has gone very high, you can see that in the panels to the right there where it's labeled, those nice red regions there, and TGF beta is essentially nil, right? EGF has won the switch. It hasn't gotten rid of the cells yet there. And if we go up higher with just transient, I know this is like just a dose of the chemical instead of having constant exposure to it. You can see if we have the low hysteresis switch, it sticks for a little bit, but then can dissolve the seam later. But if we have the high hysteresis switch, it really isn't able to ever get rid of that seam, right? And this would be what we would think would lead to palate, incomplete palate fusion and cleft palate down the road. So that's the model and some of the things that we did with it. And I will say, in leading into some of the exercises that you all can do, when we first developed the model with a single simple epi cell, right? We just had a simple epithelium across the outside of it. The palate shelves fused way too easily. We actually couldn't find a way to prevent them from fusing with one another. And so what was critical was to make sure the epithelium was polarized. And so that's what the exercises will walk you through, is thinking about making and keeping an epithelium in a model polarized. And so I hope that these .py files that are labeled here on the slide are accessible. The exercises are all about making polarized epithelia in simple systems. And so here's the first one. In the link that James is going to post, go back. Find this file, nucleatecompartments.cc3d, and follow the instructions here. And the idea is to really have differentiation built into the model. So you don't have to specify all the cells and where they go to begin with, but the cells that go to the outside of the patch then differentiate into epithelial cells, right? And the key to that, I guess the language is still the same, James, that we're talking about steppables as ways to check which cells are in contact with the medium at each step.
Yes.
And if a cell of type mesenchyme is in contact with the medium, then differentiate it to a cell of type epi with some probability. But the key is making that probability such that the outer rim of the blob, and only the outer rim, becomes type epi within about 1,000 MCS. So you'll have to play with the probabilities a little bit of that. If it's too probable or too little, you won't get enough, or you'll get too many epithelial cells.
I'm going to try to see if I can get it to run myself. It runs.
Okay, good.
Maybe I should screen share and see if what I have is what you're expecting, Shane. Is that what I should have?
No, the epithelial cells the green one should be on the outside of the patch.
Okay, well, I haven't done any exercise in terms of changing the value. This is the bare simulation as you provided.
The bare original simulation, yes, that's exactly what it should do. Just some random distribution of the two cell types.
Perfect. Okay. And the code. Let me look at the code. The code is here. And so the only thing that's happening here is that you have mitosis steppable load, I guess. Nucleate compartment steppable loaded. Okay. All right.
What we want to get rid of first is this part where it says randomly assign half the cells to be type Fe and the other half remain type mesenchyme.
Mm-hmm.
So all that stuff in the for cell in self.cell list, that entire for loop can be commented out.
Okay, remember people that there's block comment function here in the pull down menu.
Okay. Um,
Okay.
and is that going to give us, will that give us all mesenchymal cells? Let's run that and see if that gives us all mesenchymal cells. I think it
Okay.
should.
There we go.
Okay. All right.
Looks like they don't have a target volume.
They don't. Okay. Yeah. If we go back to the code, I asked you to comment out too much. We should have been a little bit more judicious in what we commented out. Um,
Okay.
yeah. Instead of the entire list, really we just want to get rid of that if random cell type equals epi. That's the one we want to get rid of, and all the other ones should be okay.
Oops. I did not block on comment. All right.
Yeah, we still need the 4-cell and self.cell list.
Thank you.
Yeah. Now it'll set a target volume for everything.
Let's see. Now, unfortunately, 4.7 seems to have a bug where when you restart, unexpected index. So I was going to show people this because we haven't really talked about it. If you have a Python bug, you'll get errors that are informative in the console. You may have to pull up the bottom line of the console a little bit to see it. And there are multiple lines. But if you look at the last line, it'll say in line 27, this is the offending code, and this is the problem. It's telling me I have an indentation error. That's very useful. Now, as I mentioned to people before, 4.7, if you try to restart too many times, it'll crash. So I'm going to close this while I'm fixing it. Let's see why there's an indentation error here. Oh, okay. No, that looks all right.
Yeah, it
I
does.
don't see an indentation error. Um,
Huh.
well, let's try again. How about other people? Is it working for you? Everyone else, somebody says, Rodrigo says the same indentation error.
Yeah.
I'm actually quite puzzled by that.
It's something to do with the line that was commented out above.
No, now
There
it's
you go.
okay.
Yeah.
So it
Yeah.
was somehow when I uncommented, there must have been an illegal character in the line. Okay, great.
All right. Okay. So that got rid of the random distribution. And so now we want to think about, okay, how do we specify so only the cells on the outside of the patch differentiate, and they do so over time, right? So instead of putting that in the start part of the steppable, we'll want to put it in the step part. And there's the instructions there for what we want to do, right? And so we can uncomment that 4-cell and self.cell list, because that's the first thing we need to do is just loop over all the cells. And the first thing we'll need to do then, when we loop over those cells, is figure out which ones are in contact with medium. What do folks think we should do to try to do that?
Somebody says cell neighbors. And I know people had asked for more demonstrations of how to use code snippets, so this is a great way of doing this. Okay, so we're going to go up to here, CC3D Python visit cell neighbors. Okay. And I didn't indent it right. There we go. Except it's not, it's a little bit, okay, that's off. All right.
Yeah, we can get rid of that line. Yeah.
Okay. Now there's something, if we want to know whether the cell is in contact with medium or not. And medium is a little bit of a strange bird in the way CompuCell does things. Neighbor has a type of none. So if I do if neighbor, then that will tell me that the cell is not medium. So if I want to know if I'm next to medium, then I have to check if my cell is of type MESENCH. And I probably should actually check if my cell is of type exists before I do that too.
Yeah.
Otherwise, I'm likely to have something fail. That's a little bit of a minefield, but they're good technical reasons why medium is actually a non-cell type. But handling it is a little bit different. So if the cell is a real cell, so this check if cell is not medium. Then we go through the neighbors, and now we say if, we can just say if not neighbor. And now check if neighbor is medium. Is this big enough for people to read? Okay. And now we're going to indent one more step.
Yep.
And now we'll say cell dot type equals self dot and MESANCH. Do people think that that should work? No guarantees.
Well, so actually we should let's see. That should be self.epi. We want to change the cell type to epi if it's in contact with medium.
Ah, okay. So let me go back and check the names. Epi here. So it should be EPI in all caps. And I only want to change it if it is of type mensch. Is that right?
Yeah.
Remember, it's a double equal sign in a comparison. All right.
Yeah,
People think
I think
that
that's not spelled correctly. I think it's
Mezenkai, boy,
MESENCH.
I'm making it Yiddish.
Yeah, and I don't know. This is just this is a programming thing that I like to do because the cell types are really just numbers. I like to early in the code somewhere just define these
Yeah, you've done
all
it
cap
here.
variables assigned to those numbers because it's easier to read the code then instead of having all these cell type 0 or cell type 1.
All right. Let me close this and reopen it so that I know that we don't crash it. Did people try this? Are people trying it? Let me know if it works when you try it. I don't think I've ever done these exercises with you, Shane.
I'm glad you know where the new things are in the code, right? How to get things done easily.
Well, he hasn't changed that much. There we go. That worked. Great.
And so now that did it right from the very beginning, like because we didn't put any probabilities, every cell that was in contact with the medium became epithelial. And you can see that we're getting a couple of double layers starting to form over time. Because if there's some fluctuations and a mesenchymal cell just gets in contact with the medium, it automatically becomes epithelial. And if we let this go for a while, we're going to get an overproliferation of epithelial cells there.
Okay, so do you now want to make this a probability rather than a definitive result?
Yep. So we'll define a variable for probability equals something. We make it a half or something like that. I would probably do it just closer to, yeah, just above that for loop.
I don't know if I have numpy random.random loaded. Something like that.
Yeah, but I think we want the if mp.random.random, I think we want that right before we assign the cell type to self.epi.
Okay.
Because we want to make sure it's under the right conditions, and then if it's under the right conditions, it probabilistically switches type.
And I think numpy is numpy.
I mean, we used random before, didn't we? Yeah, we were
Oh,
using random.random before, so.
okay. Well, then I don't need to do that. All right. I'm always used to using the numpy random function. So let's see, where was that? It was here. Okay. So we won't use the numpy one. Right. Now we'll see. I should be able to stop and restart once. So let's see if it crashes. No, it worked. Still pretty fast though.
Still pretty fast because if we had a 50% chance of it happening in every Monte Carlo step,
Right.
it's going to happen really rapidly. So we could make that, 0.1 or 0.05 as our threshold, and then it'll take a longer time.
And for people who are using 4.7 and having this crashing problem, you'll notice here at the top, initially the start CC3D was grayed out and now it's blue. That's because it takes time for it to get rid of the environment that it created in Conda. And so that's been the problem, that if you hit restart while the existing instance is there, then it crashes. And so if you actually look inside of Twit, you can see whether you're in time to reload it or not. Okay. So now it's doing a little bit more gradually. We might still want to change it to be even slower, but that's not bad.
Yeah.
All right.
But
Okay.
it's your choice with the probabilities. You can determine how fast it happens. And one of the other benefits of it happening slowly is that if there's the occasional fluctuation where a mesenchymal cell contacts the medium, it doesn't immediately change to epithelium. So you don't end up with these double layers forming and over proliferation of the epithelium. You just get that monolayer around the outside.
Okay. You want me to hand it back to you for the slides?
Sure.
I don't know if that was okay for people. That actually worked pretty well. Having you tell me what to do and am I doing it on script?
That did. I hope it worked for everybody in the audience. All right. Do folks have questions about that exercise before we go to the next one? Okay. So exercise 2 is to build on that one. All right. So the cell's differentiated into an epicell. Now, once that happens, we want it to polarize so it gets apical and basal compartments with different properties. To make this simple, we can keep the basal compartment can stay the original epicell type, but we'll need a new apical domain, epi little a, right, to represent the apical parts of the cells. The goal is to generate the polarization, maintain it, and keep the polarized epithelium well organized with all apical domains on the outside in contact with medium and all basal domains on the inside, right? So the pseudocode that we'll use to do this, we'll loop over all the cells, just as we did before, but now for each cell that's of type epi, we'll check whether the cluster it belongs to, right? We'll need to check some other part of the cell, what cluster it belongs to. Does that cluster also already have an epi-A department? If it does, well, great, that cell's already polarized. If it doesn't, then we need to form one. And forming a new compartment, right? We're taking a cell and essentially splitting it. So we have to take some part of the existing epicell and convert that to a new cell that's part of this cluster of type epi-A. So we're going to do what I call nucleate it. We'll take part of the epicell surface in contact with the medium and create a new cell for that little. You can do it as small as 1 pixel or 1 lattice site. Usually it works better to have a slightly larger area, maybe a 2 by 2 pixel area that you nucleate. And that becomes a new cell of type epi. We'll assign a few lattice sites to it, maybe 2 by 2, set its volume and surface energy parameters, and it will then grow, right? And it will expand out and become the apical compartment of the cells. One of the things you'll need to do in doing this is figure out which pixels of your cell are in contact with the medium. And you can find that function in the Palate model updated stepables that will do that.
Okay. Do you have a completed version of this as one of your is in your exercise folder?
I do not remember. Probably, but I don't remember.
Okay. The reason I'm asking is because of the way that things are organized this year, they haven't seen compartmental cells yet.
Okay.
And so I was there are a lot of other things in your exercises that would be interesting for them, but I think I was hoping we'd have the completed example for the compartments. So once you have the compartments, it's not so hard, but to figure it out the first time.
Yeah.
So let me take a look and see if we have it. If we do, then I'll load that and we can go from there.
Okay.
Okay, so people should download the Nucleate Compartments 2 file. Does everybody find that? And unzip that and run it, and that should be the one you need. That's here. And you'll notice here that these outer cells here have become green and red together. So they're compartmental here.
Yeah, and you can see just a few of them are green without a red, but a couple of more Monte Carlo steps and red should show up for those.
All right. Well, let's see.
They seem kind of resistant.
Okay, well, let me stop this one. And I'm going to I'll put in the chat the link to that file so people can download it.
Yeah. And if we just look at the code for that one, we can walk through what the various steps are doing in terms of the pseudo code we were thinking about.
And let's look at the code together. So you've defined a cell type epi and epi-A here.
Yep.
And now you have a function that finds the pixels in the next to the medium, which is actually walking through by hand and looking at each pixel up, down, left, and right.
Yeah.
I suspect we could do that a little bit more elegantly, but
yeah.
I'm not trying to be critical, but I just I suspect we could be a little more Pythonic in the way we do that.
Yeah.
Somebody could probably try to rewrite that if they're interested. Okay. And then we had talked about structure factors a little while ago the other day, that you want to make your target surface proportional to the square root of the target volume. You'll notice he's doing exactly that here. Cell.target surface is cell.target volume to the 1/2 times 4. That gives you a round cell that's not too compressed. Okay, and now here we have for cell self.cell list. You've defined a probability to differentiate of 4 out of 1,000. And now we're
step the one right underneath it, right? If you're not calling the steppable every step, if you're only calling every 10th or every 50th, right? If frequency is something besides 1, then it changes the probability. So we get the same behavior regardless of how often we call the steppable.
right. This is the Poisson relation that we're using here,
Exactly.
which is a little bit tricky. If people are not used to doing things in finite time steps where the time step changes, that conversion is always a little bit confusing to people.
Yeah.
I'm happy to talk about that during one of our discussion sessions after everything's over this today. Okay, and now we go through neighbor, get the neighbor cell list. If neighbor cell, then we're not doing anything. If we're in contact with medium, you did this in a little bit fancier the way than I did it in my code. And we change it to type epi. And now here we have a little bit more elaborate set of rules. We'll talk about these next week. Cells can be subdivided into subcells. And somebody asked about this yesterday, and we saw a little bit about it just at the beginning of it today with Gilberto. You can define a cell to have a nuclear compartment, a lamellipodial compartment, an apical compartment, a basal compartment, and that's what cluster cells are for. That defines subcells within a cell. And so cell.inventory get cluster cells will tell you what the components of the cell are. Okay? And now we're going through that list of components. And if there are no components at all, if we haven't compartmentalized the cell, this will be empty, in which case we'll return false. Otherwise, this will return true, so that we know whether the cells are compartmentalized or not. If the cell is not compartmentalized, if the cell is compartmentalized, we say that it is. If it's not, now we create a new cell ID. Self.newCell is something we haven't used much, but we have seen it in lectures before. This creates a new cell object. And now we have a list from our function of the pixels inside of our cell that contact the medium. We go through that list of pixel contacting cells. Pixel contact, medium contacting pixels. I'll get it right eventually. If there are enough of them, if there are 4, then we find a region in that, and we assign that subregion to this new cell type. This new cell ID. And then we have to reassign the properties of that new cell, both the parent cell and the daughter cell. I think you're only assigning it to the new one, so you're actually adding volume to the cell here.
Yeah.
And then if I've reassigned it, then I say that I've done it, and I continue. Okay. So this is just a taste of this. We'll go over this in detail next week. Okay. Yeah. Do you want to go now? You have another exercise you want to go over?
Nope, that was it. Those were the 2 exercises I had. If you have additional comments or suggestions, feel free to contact me or Hayden, and we can help you out with this particular code. But that was it.
And what was Palate model updated? Was that the full model?
That was the full model. It was the full model and then updated to the version of CompuCell as of 2023.
Shall we see if it runs?
Sure.
Well, I get an error when I try to run it, which I don't, not one that I know here.
Yeah, Rupla says they're getting some errors too. It'll take some debugging.
It looks like the time function isn't available. It says simulation time equals time dot clock. Time has no attribute clock. That one, is that an essential function or is that something that's optional?
I think that's optional. That's probably just in reporting, is trying to figure out how long it was taking to run the simulation.
See what happens if we comment that out.
Yeah, we might want to check and see if there's other instances of time.clock. If I was using it, I was timing things out to figure out how long different steps were taking.
All right. There. I should probably have searched for time dot clock instead of
Yeah,
just
yeah,
time.
that's probably how it showed up each time.
Yeah, you were pretty consistent about trying to benchmark your code. Uh, the problem is that I'm going to get some bugs because I'm killing these functions and big model worked for you? CMS says? That's great.
Good.
Uh,
I wonder if it's a Python version or something like that.
I wonder, yeah, it may be that I bet it's because I'm running 4.7. And I think what's loaded into the Conda environment for 4.7 may be different from 4.6. Um, how many people got to work? It's a pretty long piece of code, so
It's a lot of code there,
uh,
yeah.
you have the same error. Yeah, well, I mean, these kinds of timers, there are other timers one could use
Yeah.
um, if one wants to keep track of wall clock time. But let's see if that works. All right. Well, it started. I still have a I still have an error like that. I don't think it's terrible for people to see how to debug. Um, let's see, why can't I pull
It's
my
something everyone will have to do.
so the error is in line 871. Um, that one actually looks like I commented out something that I shouldn't have done.
Yeah.
Sell that timer. Well, I'm afraid that one I may not have time to debug. 871. Sell that timer. Okay, why is there a key error there? Did I comment out it's there? There. No, I'm not sure I'm going to be able to debug this one on the fly. Uh,
Yeah.
for people who got it to run, time might well work. Why doesn't
Yeah.
Lorenzo, do you want to try that? See if that works. But I think in the meantime, while Lorenzo is trying to get that to work, Lorenzo, did the code run for you, by the way? But this cell.timer error shouldn't be
No,
dependent on that.
yeah, that's a different error. Yeah.
Yeah. Lorenzo, do you have anything to say to add to this?
No, no, I was saying that I wasn't doing the exercise, so I haven't downloaded or tested it because I'm running other stuff. But I was looking at that. It seems that time clock is deprecated in later version of Python, apparently. And it depends also on the platform. So time, I'm sure that it works both in 4.7 and 4.6 and in latest Python.
But if you notice here, the initial condition does work. So this is another debug thing. Even if I didn't get all the way through it, you'll notice that the initial condition is displayed here. And the initial condition being displayed means that the start function works and the XML works. And so this is telling you that the bug, wherever it is in the step function. And of course, again, you come here and it says 871, and it says key error, cell.timer ecto division. And a key error means that this wasn't defined. There is no cell attribute called cell.timer ecto division. So somehow we never did that definition. Maybe that's commented out, for example, earlier in the code. But it looks like it should be there.
Yeah.
In the sense it's here. Oh, but let's go back because I didn't actually see which dictionary it was supposed to be in. Let's see, cell.timer. Is one of these supposed to be there? Perhaps. These other ones all work, huh?
Yeah. So could it have been that the dictionary for one particular cell just didn't get it set correctly?
It's possible. Sorry, I don't want to scroll and give people headaches, but 871. So it's not the key, it's maybe the dictionary. Cell attributes. So what would happen if we just comment that out? It's gonna probably clobber the logic of your code, but let's just see if we can get it to execute. Cell apoptosis flag is also not set. Also in line
872.
Yeah. So somehow here in compartment list, where we did this get pi attributes, it's not returning the information.
Yeah.
And I'm afraid that one is one that I don't know how to debug on the fly. I think this get pi attribute may not be functioning quite the same way.
Okay.
Well, I'm sorry that I don't have a solution. Okay, well, I appreciate your patience, and maybe we should put our heads together and see if we could get this debugged. This is the kind of thing where I bet ChatGPT would actually be able to fix it. Well, thank you so much, Shane. Are there any questions for Shane before you break? Those don't have to be about the code. They could be about using modeling or about computational biology or computational toxicology. Any questions? Okay, well then, thanks again, Shane. I really appreciate you coming and talking to us today.
Thanks, James.